Good morning. Thank you all for coming. It's a tremendous honor to be here today, and also a tremendous honor that our lab's work has been recognized by the Breakthrough Prize. So what I'm going to do this morning is sort of very briefly tell you why it's important that you have the number of chromosomes that you do, and what we're trying to learn by studying when you end up with the wrong chromosome number. That's that funny word, aneuploidy, that Mike just mentioned, and why it's important that we understand this. So my lab has had a long-standing interest in the molecular mechanisms that govern chromosome segregation. But what I will do today is I will tell you about our work on understanding what happens when this process goes wrong and when cells missegregate their chromosomes and end up with the wrong chromosome number. And as I mentioned, that condition is called aneuploidy. I'm showing you an example here. There is, on the left, you see a normal cell that has these 23 pairs of chromosomes. And on the right, you see an example of a highly aneuploid cell of a cancer cell. And that sort of already gives you a hint of why we believe it's important to study aneuploidy. It turns out this condition is always associated with death and disease. When chromosome missegregation happens in the cells that give rise to egg and sperms, it's actually the leading cause of miscarriages and mental retardation in humans. When chromosomes are divvied up incorrectly during the cell divisions that give rise to the body, to our soma, that is a hallmark of cancer. More than 90% of all human tumors are characterized by having the wrong chromosome number. I want to put this number into perspective for you. So over the years, people have learned a lot of mutations that cause cancer. And the most frequent gene inactivating mutation in cancer is P53. Turns out 50% of human tumors harbor mutations in P53. There's another mutation that is characterized by being hyperactive. It's a mutation in the gene RAS. And about 30% of human tumors have hyperactivating mutations in RAS. So clearly, aneuploidy is much more prevalent than any other genetic alterations in cancer. And in light of these numbers, it's perhaps not surprising, actually, that the very first hypothesis that was put forth to explain the origins of cancer was based on the observation that cancers have highly aneuploid genomes. And this hypothesis was put forth by Marcella and Theodore Boveri, who in 1914 proposed that aberrant segregation of chromosomes is responsible for a normal cell to become a tumor cell. And epidemiological studies support their idea. What I'm showing you here is a slide that demonstrates survival of patients with either tumors that have a more normal genetic complement, they are more euploid, as we say, and compare that to tumors that are highly aneuploid. And as you can see, this is an analysis, that graph up here, that shows that for all human cancers, and you can see there's a clear difference. There's also differences when we start looking at specific cancers. So my lab, a number of years ago, became interested in understanding how gaining and losing whole chromosomes contributes to tumorigenesis. But studying this question in cancer is actually quite difficult, because it turns out cancers really have messed up DNA. They don't just have whole chromosome gains and losses, they have mutations, deletions, amplifications. So figuring out exactly what it means for a cell to lose or gain a whole chromosome in the context of cancer is difficult. And so we decided to do instead is we reasoned that if we want to understand how aneuploidy contributes to cancer, we really first need to understand how the condition affects normal cells. And this is the story that I'm going to tell you about today. I will first very briefly summarize for you our work that addresses how having the wrong chromosome number affects the physiology of normal cells. And then, as per request by the Breakthrough Prize team, I will then spend most of my time talking about future challenges and what I perceive them to be. I have to give you a little bit of warning here. If you ask scientists to predict what the next big questions are, they are invariably wrong. So, because that's the beauty of science, you know? You go one way and you think you're going down this way, and then you stumble across something, and then, you know, it makes you go this way. So this is sort of what discovery is all about. But I've tried my very best to sort of come up with some ideas that I think will be important for us to address in the future. So I'm going to then first tell you about how we believe aneuploidy contributes to tumorigenesis. I will summarize some very initial, interesting, exciting data that have emerged recently in the field. And clearly, our next big question is to test these hypotheses and ideas. I will then tell you about some very recent and I believe very exciting work that suggests that aneuploid cells are actually not unnoticed by the organism. It turns out that there's actually mechanisms in the organism that clear aneuploid cells. And a key challenge then will be, of course, how then aneuploid cancer cells suppress these clearance mechanisms to develop disease. And then last but not least, I want to sort of bring up this idea of exploiting the aneuploid state of cancers as a therapeutic target. So let me briefly summarize what we've learned about aneuploidy. So we initially started our work in a model organism. It is my firm belief that studying model organisms actually provides fundamental insights into human disease. So we initially did this work in budding yeast and then later on extrapolated it to the mouse and to human cells. And so we created a large collection of both yeast, mouse, human, and human cell lines that carried specific chromosome gains and losses. They have specific names. If you gain a chromosome, it's called a trisomy. You probably have heard about trisomy 21. These are individuals who have 3 copies of chromosome 21. If you lose a chromosome, it's called monosomies. So we studied these cell lines, and then in addition, we study cell lines that actually have mutations in genes that that are responsible for segregating chromosomes accurately. So when you make mutations in these genes, what happens is these chromosomes get missegregated, and you create, pools of cells that have very highly variable and aneuploid karyotypes. So with these tools in hand, the first question that we needed to ask was, does changing gene copy number lead to a corresponding change in gene expression? Meaning, if you alter the copy number of a gene of a chromosome, will this actually affect how the RNAs and the proteins that are produced from these chromosomes are being made and their levels in cells? And in yeast and in mouse and in humans, and I believe in all eukaryotes, the answer to that question is very clearly yes. Let me show you a piece of data here. So these are yeast strains that have an extra copy of a chromosome. And what I'm showing you here is I'm arranging the genes according to their position in the genome. And then I have a method to measure the copy number of the DNA. And when it's increased, it's red, and when it's the same as it, then it's gray. So here we created these yeast strains. Each of them have an extra copy of a chromosome. And what I hope the people who are not red-green blind will appreciate here is that this leads to a corresponding increase in the production of the RNA and a corresponding increase in the production of the protein. So that result was important. The reason why this result was important is because it made us change the question that we were asking. Instead of asking, well, what happens when you lose or gain a chromosome, the real question that we had to ask is, what are the consequences of subtly changing the expression of many genes at the same time? And we spend enormous amounts of time and effort to address this question, and all my students and postdocs will be very depressed to see that I summarize all this work in basically 2 slides. So the first answer to this question is a very intuitive answer, which we call gene-specific effects. What does that mean? A gene-specific effect means that when you change the dosage of a gene, it will have consequences for the cell. You make more of it, you make less of it, the pathway may be working better now or might be working worse. The surprise came that, from a different set of phenotypes or traits that are not as intuitive, which we call the general aneuploidy-associated stresses. And what was remarkable about those is that these phenotypes or these traits are common among many different aneuploid cells, and they tend to scale with the degree of aneuploidy that exists in these particular cells. What are these phenotypes? They are basically increased free radicals, or what we call in the field oxidative stress. There's significant alterations in the metabolism, increased misfolding of proteins, and a genomic instability, meaning that they no longer faithfully copy and segregate their chromosomes accurately. What's important about these phenotypes is that they have consequences for the cells, and for the non-biologists here, really the only thing you need to remember is that they cause stresses, and the cells stop working well. Biological pathways start working suboptimally. That stresses the cells, and basically it results in a slow growth and poor proliferation. So, remember what I set out, how I set up my talk. I said, well, we want to understand how aneuploidy contributes to tumorigenesis. Well, I've just told you that losing or gaining chromosomes is actually bad news for the cells, right? They proliferate poorly. And so that conclusion is certainly at odds with the idea that aneuploidy promotes a disease that's characterized by increased proliferation and survival. And so that brings me to the second part of my talk, where I would like to sort of highlight the important new challenges. And of course, the first one is we need to understand how aneuploidy contributes to tumorigenesis. And the field has some ideas that I want to briefly summarize for you. So recall, aneuploidy inhibits proliferation. So this must mean that aneuploidy must contribute to the disease in ways other than promoting proliferation. And in this context, I think it's important to think about what actually does it mean, poor patient outcome, having less survival. Of course, high proliferative capacity is one indicator of poor outcome, but it's not the only one. We usually consider poor outcome when either the disease has metastasized from the primary site to an essential organ, such as the brain, the lung, or the liver, and when a disease has become refractory to traditional chemotherapy and other treatments. So we are starting to think that perhaps aneuploidy contributes to tumorigenesis by endowing these cancer cells with new traits. It allows them to do things that they are not normally able to do. And the reason why we're starting to think about this is because very elegant work in experimental evolution studies in microorganisms suggests that gaining and losing chromosomes can actually be beneficial under specific stress conditions. And we now have reason to believe that this is also true in cancer. I'm showing you here one very small piece of data where we're looking at the ability of aneuploid cells to survive the treatment with a very traditional chemotherapeutic called cisplatin. And in black, you see the survival of normal euploid cells, and that is less, they survive less well than, for example, these two aneuploid cell lines shown here in red. The other way we think aneuploidy contributes to tumorigenesis is by promoting tumor evolution. And that hypothesis arises from the observation, a very striking observation, that in every organism we've looked at, aneuploidy causes genomic instability. That means it reduces the ability of cells to proliferate well, to copy their DNA and segregate their DNA. We see that there's increased chromosome missegregation, increased mutations, replication stress, and defective DNA repair. And what's important about this genomic instability that we see is it's subtle. It's only 2 to 3-fold. And we hypothesize that this increase is low enough so that it doesn't cause massive cell death and kills all the cells, but it's high enough to create increased evolvability. So the ideas that the field is currently thinking about is that aneuploidy is usually an unfavorable condition, causes poor proliferation, fitness, and stress. But there are rare favorable variants that under specific circumstances allow cancer cells to do things that normal cells cannot. And then this, in combination with increased genomic instability, leads to the rapid evolution of aggressive disease. So one challenge we have is we have to test this model. These are just ideas. And a lot of people in the field are currently doing experiments to address this. The second important question that we have to address is born out of the Cancer Genome Sequencing Project. So there's a large consortium out there that sequences cancer genomes. And we were able to look at what frequency certain chromosomes are gained and lost in specific cancers. So let me orient you to this slide here. What I'm showing you here in the rows are different types of cancers. And what you see in the columns are chromosomes. In blue, you see chromosome losses. In red, you see chromosome gains. And you can very clearly see that there are some types of cancers where specific chromosomes are highly frequently gained or lost. So one example here is glioblastoma. You know, virtually every single glioblastoma has a loss of chromosome 10 and a gain of chromosome 7. So clearly, there must be something in these chromosomes that promotes tumorigenesis. And now we need to find out what they are. The next big challenge that I think we need to address is that of how cancer cells basically develop aneuploidy tolerance and allow these cells to survive despite the fitness challenges that we have. And I'm going to skip that slide in interest of time, and I'm going to just tell you that we and multiple other groups in the field have found that actually aneuploid cells are being recognized by the immune system. And of course, the key critical questions are now, what aspect of aneuploidy do these immune cells recognize? Which type of immune cells are recognizing this? And then, of course, most important, how do cancer cells that are highly aneuploid evade the immune recognition? So this is going to be exciting times learning what's going to happen. And then finally, I want to address the possibility of using aneuploidy, developing it as a new therapeutic target. We believe that such cancers aneuploidy as a therapeutic target would be highly efficacious. It would be broad spectrum. I told you most solid human tumors are aneuploid. It would also be highly specific because normal cells are not aneuploid. And several groups, including ours, including mine, have begun to look for such compounds that can do this. And we have early results suggest that they may exist. So with that, I want to thank all the many people who've worked on this project over the years. It's been a terrific experience to have worked with so many talented people. Thank you for your attention.
One of the key differences between neoplastic cells and non-neoplastic cells is not necessarily the speed at which they replicate, but the fact that when non-neoplastic cells divide, one becomes continues to be a dividing cell and the other one differentiates, so there's no net gain of cells, whereas in neoplastic cells, both become basically stem cells. So have you looked at the role of aneuploidy in contributing to this, and it doesn't really matter how fast you divide, the cells where both become stem cells will always overgrow the ones that...
So you're actually bringing up an excellent point, which is sort of the idea that that cancer cells divide really quickly. No, I actually think, no, because they don't. They don't. I understand. Let me ask let me answer my question first. Because I think there's actually a terrible misconception in the field. Because we have studied cancer cells that have evolved to divide as rapidly as possible on a dish, right, 24 hours. But if you actually look at the mitotic indices of solid tumors in a patient, they are actually exceedingly low. For example, your intestine is a very regenerative tissue. At any given time you look at that tissues, about 20% of those cells will be in mitosis. In a primary breast cancer, that number is somewhere between 1% and 2%, which is actually the reason why you die of cancer in your 70s and not when you're 12. So you don't, basically they're dividing. I think the key to cancer is more like they're dividing when they're not supposed to divide. They're doing a bad job at it, but they basically escape the controls that keep these cells in G1. I don't think, what I'm trying to say is you don't even have to evoke a fate transition that they become more stem-like or not stem-like. I think the only thing you have to invoke is that they have sort of basically escaped the G1 arrest control that most terminally differentiated cells are underlying. That's what I tried to say.
Thank you for bringing importance of aneuploidy in this platform. My question is, what do you think? Is aneuploidy is a cause or is a consequence of the cancer?
It's an excellent question.
What can cause aneuploidy?
So you're bringing up an interest. So I actually personally think it's both. You're bringing up an interesting question, which is sort of what comes first? Are there oncogenic mutations that then lead to aneuploidy, or is aneuploidy first? So I personally believe is that the oncogenic drivers are initiating tumor genesis, and that aneuploidy then basically helps the cancer to develop and evolve and do the things that moving from simple hyperplasia to aggressive disease. That all our data suggested. However, we've actually recently developed a mouse model where we, to my surprise, and that's why I'm saying never predict anything for 10 years. So we just recently developed a mouse model of cancer, aneuploidy that develops cancer with exceedingly high penetrance. And we actually understand why all the other mouse models that people have developed previously didn't show a much increased tumor genesis. So I think it can be both.
Do you have any ideas why some organisms seem to handle polyploidy sort of just fine? Oh, sorry. Any ideas?
There's a big difference between polyploidy and aneuploidy, and I apologize. I should have explained that better in the beginning. Polyploidy means that your entire genome gets doubled. And all the plants that we personally care about are basically polyploid, right? So what's key about polyploidy is that the relative dosages of genes remains the same. And what happens in aneuploidy is that these relative ratios of genes are altered. And that's what causes problems. There used to be some literature that suggests that some organs, even in our bodies, are aneuploid. That's just not true. The good news is all your cells are euploid, and even if you have some aneuploid cell, the immune system will kill it. So thank you very much.